Hello, everybody. Thank you for joining my talk today. My name is Rachita Sumbria, and I'm an associate professor at Chapman University School of Pharmacy and also an associate project scientist at UC Irvine. For almost 2 decades, I've been studying the blood-brain barrier, and for about last decade or so, we've been focusing on using the transferrin receptor-mediated approach to deliver biologics across the blood-brain barrier for Alzheimer's disease. And that's exactly the title of my presentation. As you can see, I'll be talking about the use of transferrin receptor-mediated drug delivery for Alzheimer's disease. So let's get you all started with a little bit of a background about Alzheimer's disease. And Alzheimer's disease is the leading cause of dementia in the United States, and we have close to about 7 million Americans aged 65 years and older living with Alzheimer's disease. And sadly, this number is expected to increase to about 14 million by 2060. And the main reason driving this increase in the cases for Alzheimer's disease is that we have an aging baby boomer generation, and we also really do not have a cure for Alzheimer's disease. And if you were to look into a brain of a patient with Alzheimer's disease, as you can see in the figure to my right here, what you see in the brain of this patient with Alzheimer's disease is that you have a lot of amyloid beta accumulation, which is basically found outside the cells or extracellularly. And along with that, you also have something called neurofibrillary tangles, which begin to deposit within the neurons. And besides A beta plaques and neurofibrillary tangles, we also have a large amount of neuroinflammation, microglial activation that's ongoing, and which eventually leads to neurodegeneration, leading to AD dementia. So you see that there are key hallmark pathologies associated with Alzheimer's, and this has eventually led to 3 disease-modifying therapies, out of which 1 was withdrawn, but now 2 disease-modifying therapies for Alzheimer's disease that were recently approved by the FDA just in the last 2 years. And both these therapies target the accumulation of amyloid beta plaques, which are these large brown accumulations that you see in the figure. So this is an exciting time because now we eventually have 2 disease-modifying therapies for Alzheimer's disease, but still there is more work that needs to be done, which can focus on other aspects of the disease, for example, neuroinflammation and neurodegeneration. So keeping that in mind, if you look at the progression of various biomarkers for Alzheimer's disease, if you look at this particular figure here, it very nicely shows you the progression of these AD biomarkers, specifically focusing on the purple and the red curves, which are the first 2 curves in the graph here, which show you the amyloid beta accumulation in the brain. And if you look at the green region towards the right of the curve here, it shows you the progression of AD dementia. What you notice is that the amyloid beta plaques, which are shown in the purple and the red, begin to accumulate many, many years, maybe even decades before any symptoms of Alzheimer's become obvious in these patients. And the field largely believes that it's after amyloid beta accumulation that tau follows, which is the light blue third curve here. And these 2 are the major hallmark pathologies, as I explained on the previous slide. But apart from tau and amyloid beta progression shown in this graph here, you see there's a brown curve, which is very peculiar and is really not following the trajectory shown by the other biomarkers. And this brown curve here basically signifies microglial activation. And the microglial cells are the immune cells of the brain. So what you notice with respect to microglial activation is that this curve really rises very steeply early on, even before a person may know that they have Alzheimer's disease, and then begins to plateau when AD dementia starts progressing. And just besides microglial activation, which is happening in the brain, there are other important factors that are happening in the periphery. For example, we have systemic inflammation, we have obesity, we have other risk factors which collectively increase systemic inflammation. So it's really not just the brain inflammation associated with microglia, but also systemic inflammation, which appear to be really early events in the process of AD pathogenesis. So keeping this in mind, inflammation therefore becomes a really important target for the disease. And when anyone talks about neuroinflammation or inflammation in general, TNF alpha is a key player in that regard because it is a major pro-inflammatory cytokine. And specifically with respect to Alzheimer's disease, there's collective evidence today showing us that TNF levels are elevated in the serum of patients with Alzheimer's disease. Also in postmortem brain tissues, you see a lot of accumulation of TNF alpha associated with amyloid beta plaques, and collectively TNF levels are associated with AD progression. So when you start thinking about this and you're like, okay, well, TNF is an important target for the disease, then can I develop TNF inhibitors for Alzheimer's disease? And the question is absolutely yes. And the good thing about this is that we already have FDA approved TNF alpha inhibitors for other inflammatory conditions, for example, for Crohn's disease or rheumatoid arthritis. And all these TNF inhibitors are biologic or large molecular weight drugs. Now, the issue with trying to repurpose these drugs for Alzheimer's disease is that we have to bypass the blood brain barrier, which pretty much blocks 98% of biologics from getting into the brain. So, in order to overcome this challenge, researchers have, directly injected these biologic inhibitors into the brain or through the intracerebroventricular route to try to get them into the brain. But obviously, those routes are not, clinically applicable. So, in order to overcome this challenge, this is where our research, becomes important. Is that we decided to use drug delivery strategies to shuttle these biologic TNF alpha inhibitors into the brain for Alzheimer's disease. And the use of blood-brain barrier transferrin receptors for drug targeting to the brain is not novel. This was a concept, or this was an idea that was suggested about 40 years ago by Bill Partridge's group, who basically showed that this approach could be effectively utilized to deliver biologics to the brain. And it's a very simple approach. If you look at the cartoon to the right here, what is done in this approach is that you're basically targeting the transferrin receptors, which are overexpressed at the blood-brain barrier. And what you can do is that you can design antibodies that target the transferrin receptor. And you have your antibody on one end, which is then fused or essentially attached to your biologic drug of choice on the other end of the antibody. So in theory, what happens is when you inject this fusion protein, which has the antibody on one end, and your biologic drug on the other end, when you inject this systemically, this fusion protein will then bind to the blood-brain barrier transferrin receptor, which is known to be a receptor-mediated transcytosis system, meaning it will move the cargo from one side of the cell to the other side of the cell, in this case, effectively delivering it to the brain. So just to give you a little bit of historical perspective about how the transferrin receptor today is the most widely studied target for brain drug delivery. In the early 1980s and 1990s, blood-brain barrier transferrin receptor expression was confirmed, and it was shown that these receptors are enriched at the blood-brain barrier, and that this receptor could mediate receptor-mediated transport. And then in 2000 to 2010, antibodies were then shown to cross the blood-brain barrier that were targeting the transferrin receptor. And then linker chemistry was used to attach drugs to these antibodies. And it was shown that this approach could effectively deliver the biologic to the brain. And this approach was then further sophisticated by using recombinant technology, where fusion proteins were engineered, where you had the antibody on one end and your biologic drug of choice on the other end. And really, from 2010 to present, there's been a lot of interest in using this approach, not only for the blood-brain barrier transferrin receptors, but other receptors as well to deliver biologics into the brain for Alzheimer's disease. And quite excitingly for the field, we do have a market approval where transferrin receptor antibody is used to deliver a therapeutic enzyme to the brain, and this was approved in Japan. And currently, there is an antibody being developed by Roche where you have the Fab fragment of the transferrin receptor antibody attached to an anti-amyloid antibody being developed for Alzheimer's disease. So hopefully this gives you a little bit of a historical perspective on where we are with respect to the transferrin receptor-mediated drug delivery approach for brain drug delivery. So using this approach, we then decided to now attach our biologic TNF alpha inhibitor, which is basically the extracellular domain of the TNF receptor, which is shown in the cartoon to the left. So we have the transferrin receptor antibody, which is now attached to the TNF receptor. The TNF receptor domain is what acts as the biologic TNF alpha inhibitor by attaching or binding to the TNF, sequestering the TNF and preventing any downstream TNF signaling. So once we had this fusion protein, we then confirmed that it retains its bifunctionality. That is, it should bind to the transferrin receptor, which is what we showed in the top curve here, where we showed that the KD was in low nanomolar range for binding to the transferrin receptor. But we also confirmed or had to confirm its binding to the TNF alpha. And the lower curve shows you that the KD for binding to TNF alpha was also in the low nanomolar range, which basically confirmed that this fusion protein retained high affinity binding to both its targets, that is the transferrin receptor and TNF alpha. We obviously then wanted to check the biological activity of this fusion protein to make sure that it's functioning. We did this by several approaches, but the data that I'm showing here shows you a cell culture system where we made use of iPSC-derived brain microvascular endothelial cells, and we measure the TR values, which shows you how intact the monolayer is. And if you look at the black curve in this graph in the middle here, you see that the TR values are maintained, which is just your cells without any TNF treatment. However, as soon as we add TNF alpha, we see a decrease in TR, telling you that the monolayer becomes leaky. However, as soon as we add our fusion protein, which is the blue curve, you see the restoration of TR values, telling you that our fusion protein was able to neutralize the effects of TNF alpha and that it is biologically active. It was also confirmed that this fusion protein does enter the brain past the vasculature, which was important to show that it does get into the brain tissue and passes the blood-brain barrier. And as you can see, the blue bar to the right here shows you the total volume of distribution of the fusion protein in the brain homogenate, and the red shows you what's actually in the brain parenchyma, and 80% of the fusion protein was actually found in the brain parenchyma, confirming the effect of transcytosis of this particular molecule. So after these initial studies, we then performed the first proof of concept study to see if this fusion protein, which is a brain-penetrating TNF inhibitor, is this protective in a mouse model of Alzheimer's disease. So we use a very simple approach. We use an amyloid beta mouse model, which is the APPPS1 mouse model, which overexpresses human amyloid precursor protein. And we injected our brain-penetrating TNF alpha inhibitor into these mice for 3 months. And these mice were 9 months old when we sacrificed them. So apart from just having a group that was treated with just saline, which is a control group, and a group that was treated with a brain-penetrating TNF inhibitor, we also had an additional group that was treated with etanercept, which in theory is a TNF alpha inhibitor, but is not expected to cross the blood-brain barrier because it does not have the transferrin receptor antibody attached to it. So our hope was to see if our brain-penetrating TNF inhibitor was more effective than etanercept, because our brain-penetrating TNF alpha inhibitor could transverse the blood-brain barrier. So as you can see from the confocal fluorescent images, both in the cortex and in the hippocampus, we saw visually a lowering of our amyloid beta plaques in the brain-penetrating TNF alpha inhibitor group. And when we quantified these, the data is shown in terms of the bar graphs, where we quantified the total 6E10 positive area and also the number of punctures or plaques that we saw. And as you can see from the bar graph, it was the brain-penetrating TNF alpha inhibitor treated mice that had lowering of amyloid plaques. However, our etanercept treated mice did not show a significant lowering, though there was a slight trend, but not statistically significant. We also followed it up by specifically measuring the levels of brain A beta 142, which is considered to be the more neurotoxic or pathologic species of amyloid beta. And as you can see in the bar graph to the right here, we again saw a significant lowering with our brain-penetrating TNF inhibitor treatment, but not with etanercept, again confirming that yes, delivering the biologic TNF alpha inhibitor by the transferrin receptor antibody was more effective than in the absence of it. So we did collect other data in that study, but for the sake of brevity, I'm going to move on to the second study that we conducted after the first study. And the aim really was, after seeing these promising effects in the 9-month-old APPPS1 mice, we asked the question of whether, if we could delay the treatment in these mice, say we started treating these mice when the amyloid pathology was fully blown, could we still reverse it, right? So delayed treatment of this inhibitor, is it still effective, was essentially the question we were asking. So we used exactly the same mouse model. However, we started treating this time when the mice were 10 months old, and we sacrificed the mice when they were 13 months old. So basically delaying the pathology. And we were quite surprised by the efficacy of this therapeutic approach. As you can see from the bar graphs, both in the cortex and the hippocampus, the purple curve is the one that was basically the mice treated with the brain-penetrating TNF inhibitor. And you see there was a dramatic, significant reduction in the amyloid load with our brain-penetrating TNF inhibitor. If you focus your attention onto the pink bars there, which basically shows you the amyloid deposition with etanercept, which is not supposed to cross the blood-brain barrier, we really did not see any lowering in amyloid plaque. And we had an additional treatment group in the study where we just added the transferrin receptor antibody alone, just to make sure that the antibody alone was just to understand if the antibody alone was having any effects. So this was actually very encouraging because now we had delayed the treatment and we still saw significant lowering with our brain penetrating TNF inhibitor, and etanercept really did not do that. So we did see encouraging results with amyloid beta lowering, but do we see any improvement in memory was the next question we asked in the same study. So we performed the Y-maze test, which is a spatial reference memory test, which is basically relying on the ability of the mice to explore a novel arm more than a familiar arm. And supporting our amyloid beta results, what we found was that it was a brain-penetrating TNF alpha inhibitor treated mice, which is a purple bar graph here. Those mice actually spent more time in the novel arm, traveled greater distance in the novel arm, and also had reduced latency to enter the novel arm. All parameters telling us that the spatial reference memory was significantly enhanced or improved in these mice that were treated with a brain-penetrating TNF inhibitor. However, the mice that were treated with just the antibody alone, which is the orange bar, or just etanercept alone, which is the pink bar, really did not show any improvement in spatial reference memory. We also noticed that the mice that were treated with a brain-penetrating TNF inhibitor had a greater tendency to select the novel arm as their first arm choice, which is shown in panel D here, again confirming that yes, there was improved spatial reference memory in these mice with a brain-penetrating TNF inhibitor and not with a non-brain-penetrating TNF inhibitor. So the other areas that we looked at in this particular study was trying to understand how the TNF inhibitor may be working. And one area that we looked at was looking at the blood-brain barrier protection that may be offered by the brain-penetrating TNF inhibitor. And panel A and panel B, we measured ZO1 and Claudin-5, which basically tell us about the blood-brain barrier integrity. And we found that both ZO1 and Claudin-5 was significantly increased with our brain-penetrating TNF inhibitor. So this was great because we were seeing great efficacy measures with our brain-penetrating TNF inhibitors, but we also have to consider certain aspects while dealing with the transferrin receptor antibody. And the key one being that transferrin receptor antibody chronic dosing is shown to be associated with anemia, primarily because the reticulocytes, which are the immature red blood cells in mice, have a high expression of transferrin receptor, and the transferrin receptor antibody somehow causes lysis or loss of these reticulocytes. So an important parameter for us to consider was look at hematologic indices in these mice to see if those were altered with our treatment. And we were very pleased with the results because if you look at the purple bar here, which is a brain-penetrating TNF alpha inhibitor mice, we really did not see any change in hematocrit, hemoglobin, or MCV values, but we did see an alteration with just the antibody alone, which is the orange curve, orange bar, which we actually expected going into the study because it's a known side effect of just using the transferrin receptor antibody alone. So this just tells us that looking at the transferrin receptor antibody alone does not give you a complete picture of the side effects that may be associated with the use of that approach. Because as soon as we fuse the transferrin receptor antibody to the TNF alpha receptor, the safety profile completely changed. In fact, the fusion protein had a much better safety profile with just compared to just the antibody. That's why it's important to consider every molecule on a case-by-case basis and not just use the behavior of the transferrin receptor antibody to predict side effects of by using the transferrin receptor antibody as a drug delivery approach for brain diseases. The other important question was, because the transferrin receptor antibody targets the transferrin receptor, what happens to iron homeostasis? Are you keeping that in balance? Are you changing that because of the chronic dosing in these Alzheimer mice? So we measured the levels of different iron receptors. We measured transferrin receptor and ferroportin, which is an efflux transporter for iron. And we did this in the brain, spleen, and the liver. And we really did not see any change in the levels of transferrin receptor expression or ferroportin levels in the brain, liver, or spleen with a brain-penetrating TNF inhibitor. We did see an elevation in the levels of transferrin receptor and ferroportin in the spleen with just the transferrin receptor antibody alone, which is an expected finding, something that we've also previously reported, but we really did not see that with the brain-penetrating TNF inhibitor, again underscoring that the attachment of the fusion partner to the antibody changes how the molecule behaves. We also measured the levels of iron in the spleen and the liver just to make sure that our brain penetrating TNF inhibitor was not altering that. And we really did not see any changes in spleen iron or liver iron as shown in panels J and K with our brain penetrating TNF inhibitor. But again, we did see an elevation just with the antibody alone, again telling us that the fusion protein had a much better safety profile than the antibody alone. So this was great. So we had studies done in two different mouse models of amyloid beta, not two different, but one mouse model of amyloid beta, but then we wanted to follow it up and see the effects of this fusion protein in a tau model, because that is one of the other hallmark pathology of Alzheimer's disease. And we decided to do this in PS19 mice, which is a very popular mouse model of tauopathy. The results were quite interesting and very different from what we had seen with the APPPS1 mice shown in the previous slides. So here is when we started getting into sex differences, because this was the first time we had male and female mice in our study. And the top panel here shows you phospho-tau accumulation in the male mice, and the bottom panel B shows you the phospho-tau accumulation in the female mice. So if you focus your attention onto the blue bar in this figure here, which shows you the mice that were treated with the brain-penetrating TNF inhibitor, in the top panel, you really do not see any change in the phospho-tau accumulation in male PS19 mice. However, we did see a significant lowering with etanercept. And remember, etanercept is the one that does not have the antibody fused to it. So we really did not expect this finding going into the study. But when we looked at the female mice, the results were even more puzzling, where we found that the brain penetrating, which is the blue bar, and the non-brain penetrating, which is the orange bar, both of these mice had a significant lowering in phosphorylated tau. So this told us there are some sex differences going on here with respect to tau pathology. The brain-penetrating TNF-alpha inhibitor is not so effective in the male mice, but effective with the female mice. And in fact, etanercept seems to be doing better in this tauopathy mouse model. So at that point, we had several hypotheses running in our heads as to what may be going on, and we were kind of leaning towards the idea that maybe tauopathy and TNF alpha are associated differently than amyloid beta pathology and TNF alpha. So we said, let's combine the two pathologies in using a human tau and A beta mouse model, and we use a triple transgenic mouse model of Alzheimer's disease to see what happens to amyloid beta load and tau pathology in this particular mouse model. And if you look at the first bar graph here, we did see a significant lowering in amyloid beta pathology, which is consistent with what we had seen in the APPPS1 mice. However, if you look at the AT8 or the tau pathology graph to the right here, we really did not see any movement or any reduction in tau pathology with our brain-penetrating TNF-alpha inhibitor. Again, this was indicating that, tau pathology is behaving differently with the inhibitor versus amyloid beta pathology. So we said, okay, let's follow it up with another study where we escalate the dose, because we thought maybe the dose wasn't enough to move tau pathology in these male PS19 mice. And we said, okay, let's up the dose and see what happens. So we performed a dose escalation study in the same mouse model, which is the PS19 mouse model. And we increased the dose from 2 milligram per kilogram to 5 milligram per kilogram to see if that really helped change the pathology in male mice, because female mice already were showing an effect. And this was very interesting, because when we upped the dose, as you can see, there was a significant reduction in the tau pathology in the male mice, which is a red bar here, with the brain-penetrating TNF inhibitor. However, now with the increase in dose, we saw a worsening in pathology in the male mice, which is the red bar, as you can see in the female bar graph. So we were able to improve the pathology in the male mice by increasing the dose, but somehow we landed up worsening the pathology in the female mice. So at this point, we think that there's a tipping point to how much TNF inhibition is tolerated or beneficial in female mice, but in the male mice, clearly increasing the dose was beneficial. So we started digging more, and we wanted to find out other reasons that could potentially be contributing to these sex differences. And we said, okay, let's look at the transferrin receptor expression, because the whole efficiency of this strategy really depends on the expression of the transferrin receptors. And if those are altered, of course, that probably could explain what was happening in the male and the female mice. So we measured transferrin receptor expression in the liver, in the spleen, and the kidneys, and we did not see any sex differences. So we combined the male and female mice for these organs. We really did not see any changes with the brain-penetrating TNF inhibitor, which is a black bar here. We did see a lowering in the spleen, in the liver with our just the antibody alone, which is telling us that higher doses of the transferrin receptor antibody alone does result in a reduction in transferrin receptor expression in the liver. However, our fusion protein did not alter these levels in the liver, spleen, or the kidney. Interestingly, when we dug into the plasma levels, what we found was that we saw significant sex differences. We saw that the female mice had significantly lower plasma transferrin receptor levels compared to the male mice. And then we asked ourselves a question: if this is happening in the plasma, is this also happening in the brain? And when we studied the brain tissue and when we looked at the transferrin receptor vessel count using immunostaining, we found that yes, the female mice indeed had lower transferrin receptor expression than the male mice. So basically, we ended the study knowing that there were sex differences in efficacy and there were also sex differences in the transferrin receptor expression. And both of these factors could significantly impact the way that this molecule behaves with respect to tauopathy. So this brings me to my conclusion. So from the 4 studies that we've done, actually 5 studies that we've done with the brain penetrating TNF inhibitor, what we can conclude is that the brain penetrating TNF alpha inhibitor does result in better therapeutic indices than etanercept alone in young and aged amyloid beta APP-PS1 mice. Very robust effects in these mice. However, we really did not see the same effects in the tauopathy mouse model of where we had human tau overexpression. What we also found was that the fusion partner regulates the side effects mediated by the transferrin receptor. And we also found significant sex differences in the effects of the brain penetrating TNF alpha inhibitor in the PS19 tauopathy mice. And we also found significant sex differences in the transferrin receptor expression in the PS19 mice, as shown on the previous slides. So there is a lot of work that still needs to be done with all this data that I've presented. This is really not complete. It's not giving us a complete picture of what's going on. There are several areas that we need to consider. For example, we have to further evaluate sex differences in the plasma and the blood-brain barrier transferrin receptor expression in AD mice if we have to use a transferrin receptor antibody-mediated approach for drug delivery, because that is clearly going to impact how well this treatment strategy works. We also have to further understand the impact of chronic dosing of the transferrin receptor antibody fusion protein and how that impacts the bioavailability of the fusion protein. And what are the mechanisms that regulate the bioavailability and the safety profile of the transferrin receptor fusion proteins. And just specifically with respect to TNF alpha, it's also important to consider sex differences because it is known in the field of rheumatoid arthritis and where TNF alpha inhibitors have been used for a long time, that females are less responsive to therapy than male mice. So there are 2 factors we have to consider here now: the transferrin receptor sex differences and also the TNF alpha mediated sex differences, which are at play while trying to develop this particular fusion protein. So I will stop at that, and I have a number of people I would like to thank who've contributed significantly to this work over many years. A number of postdocs, number of graduate students have contributed significantly, and I highlighted them on each of the slides. My collaborators at Pomona College and UC Irvine, and of course, my mentor, Bill Partridge and Ruben Boado, who introduced me to this technology, and of course, the funding that was provided to continue with this work, without which this work wouldn't be possible. And thank you all for listening to me.